As the HGP lurched into high gear, the debate persisted about the best way to proceed. Some pointed out that a large portion of the human genome is what we in the trade call junk, stretches of DNA that apparently don't code for anything. Indeed, those stretches that encode proteins, genes, constitute only a small fraction of the total. Why, therefore, these critics asked, should we sequence the entire genome? Why bother with the junk? There is actually an extremely quick and dirty way to secure a snapshot of all the coding genes in the genome using the reverse transcriptase technology described in Chapter 5. Purify a sample of messenger RNA from any type of tissue. If your source is the brain, you will have a sample of RNA for all the genes expressed in the brain. Using reverse transcriptase, you can then create DNA copies, known as cDNAs, of these genes, and the cDNAs can then be sequenced. This quick and dirty approach, however, was no substitute for doing the whole thing. As we now know, many of the most interesting parts of the genome lie outside genes, constituting the control mechanisms that switch the genes on and off. And so in the case of the cDNA analysis of brain tissue just described, you'll have an overview of the genes switched on in the brain, but no idea how they are switched on. The hugely important regulatory regions of DNA are not transcribed into RNA by the RNA polymerase enzyme that copies the DNA strand into messenger RNA. Working at the relatively cash-strapped Medical Research Council, MRC, in Britain, Sidney Brenner pioneered this cDNA-based approach to large-scale gene discovery. With limited research funds, he figured that sequencing cDNAs was the most cost-effective way of using what little money he had. Keen to reap the commercial benefits of the sequences, the MRC prevented Brenner from publishing them until British pharmaceutical firms had a chance to position themselves to profit from them. On a visit to Sidney Brenner's lab, Craig Venter was impressed by this cDNA strategy. He could hardly wait to return to his NIH lab outside Washington, D.C., where he would apply the technique himself to produce a treasure trove of new genes. By sequencing even a small part of each one, Venter could determine whether or not it was new to science. In June 1991, an NIH official urged him to apply for patents on 337 of these new genes, although he had, in many instances, no clue about their function. A year later, having applied the technique more broadly, Venter added 2,421 sequences to the list submitted to the Patent Office. In my judgment, the very notion of blindly patenting sequences without knowledge of what they do was outrageous. What, precisely, was one protecting? This conduct could only be seen as a preemptive financial claim on a truly meaningful discovery someone else might yet make. I expounded my objections to the higher-ups at NIH, but to no avail. And the agency's persistence in endorsing the practice, a policy that was later reversed, spelled the beginning of the end of my career as a government bureaucrat. I had mixed feelings when Bernadine Healy, head of NIH, forced me to resign in 1992. 4 years in the Washington pressure cooker had been enough. But what really mattered to me was that by the time of my departure, the Human Genome Project was undeflectably on course. Venter's taste of the commercial possibilities of patenting chunks of the genome whetted his appetite for more. But he wanted it both ways, to remain a part of the academic community in which information was freely shared and salaries were small, and also to enter the business arena in which his discoveries could be kept under wraps until the patent cleared and he could cash in. With the help of a fairy godfather, venture capitalist Wallace Steinberg, the inventor of the Reach toothbrush, Venter got his wish in 1992. Steinberg supplied $70 million to set up not one but two organizations, a nonprofit, the Institute for Genomic Research, TIGR, pronounced Tiger, to be headed by Venter, and a sister company, Human Genome Sciences, HGS, to be headed by commercially inclined molecular biologist William Hasseltine. It would work this way. Tiger, the research engine, would crank out cDNA sequences, and HGS, the business arm, would market the discoveries. HGS would always have 6 months to review Tiger's data prior to publication, except when the findings indicated potential to develop a drug, in which case HGS would have a year. Having grown up in California, Venter initially chose surfing over higher education, but a traumatic year-long tour as a medical assistant in Vietnam during the war seemed to focus his mind, and on his return to the United States, he acquired in short order an undergraduate degree and a PhD in physiology and pharmacology from the University of California, San Diego. His migration from academia into the commercial sector made sense, viewed in relation to his personal finances. By his own reckoning, he had $2,000 in the bank when he founded Tiger. But he was quick to turn his fortunes around. Early in 1993, the British pharmaceutical company SmithKline Beecham, anxious for a stake in the genome gold rush, paid $125 million for the exclusive commercial rights to Venter's growing list of new genes. And a year later, the New York Times revealed that Venter's 10% share of HGS was itself worth $13.4 million. Not afraid to spend it, he dropped $4 million on an 82-foot racing yacht, whose spinnaker he adorned with a 20-foot image of himself. In the 1970s, William Hasseltine had been at Harvard as a graduate student under the joint direction of Wally Gilbert and myself. Afterward, he would run an innovative HIV research center at the medical school's Dana-Farber Cancer Center. But it was his marriage to the multimillionaire socialite Gail Heyman, creator of the 1980s must-have perfume, Giorgio Beverly Hills, that gave him the most visibility and ensured Hasseltine had rather more than the $2,000 in the bank when he set up HGS. Even before he went corporate, his jet-setting had provoked comment from members of his Harvard Medical School laboratory. What's the difference between Bill Hasseltine and God? Answer: God is everywhere. Hasseltine is everywhere but Boston, where he's supposed to be. Precious little skill or ingenuity was involved in Venter and Hasseltine's scramble to patent every human gene they could find on the basis of cDNA sequencing. Tiger and HGS were simply the biotech equivalent of the kids who round up all the toys at the playground just so no other kid can play with them. In 1995, HGS filed a patent for a gene called CCR5. HGS's preliminary sequence analysis suggested that the gene encoded a cell surface protein in the immune system and was therefore worth owning, since such proteins may potentially serve as targets for drugs affecting the immune system. CCR5 was one of a batch of 140 patents for similar genes that HGS applied for. But in 1996, researchers discovered the role of CCR5 in the pathway by which HIV, the virus that causes AIDS, invades the immune system's T cells. They also found that mutations in CCR5 were responsible for AIDS resistance. It had been observed that some gay men who turned out to have mutated CCR5 genes never contracted the disease despite repeated exposure to HIV. Thus, CCR5 was and remains clearly destined to play an important part in our assault on HIV.